like that will again be fairly descriptive, not real quantitative, etc. Please remember that when we're done with this chapter, we will have our second hour exam. Before we have that, we will have the sample exam, and then we'll do the real thing. One other thing, in the lab schedule, they put in 2 or 3 days that we could use for catch-up. So if we're behind a lecture, or if they cancel the lab for some reason, whatever. We are not behind. We're actually doing very, very well. Therefore, on Monday, we will have one of these days, and there is no lab. So please don't come. I won't either. I will send everyone an email just to make sure that we all get the message. Any questions? Well, let's start off with the concept of valence. We've talked about this before. Valence is simply a word that describes how many bonds there are to various atoms in their ground state. Now, when we say ground state, this is their common, the way you commonly find them, okay? Not charged, anything like that. We know that oxygen, as in water, will form 2 bonds. It has 2 unshared pairs of electrons. Nitrogen will form 3 bonds, and it has 1 unshared pair. Poor boron forms 3 bonds also, but it has no unshared pairs, so it does not have the octet. Remember, this makes boron compounds very good electron acceptors because they desperately want another pair. And finally, carbon. Carbon has 4 bonds in virtually all neutral carbon compounds. Today we're going to be talking about compounds of carbon. Now we know that carbon is in the second period. Atomic number is 6. Because it's in the second period over here on the periodic table, that means that the 1s, the first level, is totally full. We have 2 electrons in there. The 2s is also filled. And we have a total of 4 electrons in our second level. So these must be placed singly in the p orbitals. Our total configuration looks like this, or 1s2, 2s2, and 2p2. As we said, carbon forms 4 bonds. So in order to form 4 bonds, we actually have to split up these electrons here in the 2s, and we're going to do what's known as a hybridization. We're going to take the 2 electron, or one of the electrons out of here, going to move it down here to a p orbital. So we have 4 electrons that we can use to make bonds, and that gives carbon our valence of 4. Now, according to the valence shell electron pair repulsion theory, we would expect something that forms 4 bonds to be tetrahedral. And in fact, carbon is. This is methane. It's the very simplest of carbon compounds. 1 carbon in the middle and 4 hydrogens. This is tetrahedral geometry. That means that all of these angles are all the same, 109.5. All the distances here are exactly the same. We can get rid of the model. You can see the tetrahedron a little clearer this way. It's basically a pyramid, except the carbon here is not in the base. It's moved up in the middle of all of these atoms. This is what carbon looks like. And again, all compounds of carbon that have 4 bonds to it will be tetrahedral like this. We made this by taking one electron from the s orbital, and we put it up in the p orbital. So we're actually constructing our bonding orbitals from one s and three p's. In organic chemistry, we refer to this as a hybrid. And just so that you know the nomenclature, this is an sp3 hybrid. Again, one s orbital, three p orbitals, and we arrange the bonds so that they are tetrahedral. Everything is as far apart from each other as it can get. And again, 109.5 degree bond angles. sp3 hybrid. A little bit later, we'll see an example of an sp2 and an sp. So those are different ways that we can bond to carbon. Now, the neat thing about carbon and carbon compounds is that we can connect these carbons together. We can connect them in very, very interesting ways. We can stick in oxygens, nitrogens, sulfur, other things along with our chain. This is a functional, or in organic chemistry, we speak of functional groups. This would be an alkane. An alkane contains only sp cubed hybridized carbons. So every carbon in our chain here is going to be tetrahedral. Functional groups are actually really important in organic chemistry. We'll talk about these later, and we'll look at lots and lots of them. The interesting thing about a functional group is that it makes organic easy, believe it or not, because functional groups will have characteristic reactions. And they're pretty much independent of what the actual molecule looks like. You just look for the functional group. But okay, so this is an alkane, it's called. Contains only sp3 hybridized carbons. Because all of these are tetrahedral, this thing actually looks like this. So these are tetrahedral carbons. This zigzag structure, we'll see that a lot. Whenever we are drawing carbon compounds, typically we will use a zigzag to represent the chain. That's just an attempt to show what the geometry actually is. In addition to nice straight chain alkanes, we can also make them a little more interesting. Here we've taken our straight chain and we've put another carbon attached to it. Call this a branch chain. Again, this is an alkane, so everything here is tetrahedral, and what it looks like is actually something like this. Here's the extra CH3 that we just stuck in, and again, here's our basic zigzag with the CH3 now sticking up. Branch chain alkane. Any questions? When you have the functional group of an alkane, the general formula will always be given by this little equation. Cn and H2n plus 2. So n is any integer. Here we have 1, 2, 3 carbons. So n would be 3. 2 times 3 is 6. Plus 2 more is 8, so we have a total of 8 hydrogens. C3H8, again with N equal to 3. Over here we did a branch chain. We have 1, 2, 3, 4, 5 carbons. This is C5H12. Again, our equation here with N equal to 5. Now we can arrange these chains of carbon atoms in lots and lots and lots of ways. When we're dealing with an alkane, we're going to name these things based on the number of carbon atoms, and we're going to add the suffix ane to indicate that this is an alkane. So if we have one carbon, as we said earlier, this is CH4. One in organic talk is meth, M-E-T-H, and we simply stick ane in here to show that it's an alkane. 2 is eth, E-T-H. 3 is prop, but, pent, hex, hept, and these are longer. So, you know, these are the ones that are common. Butane, 4 carbons, C4H10. Again, they all follow the basic formula, CnH2n plus 2. We had a straight chain the other day, or in the other slide, and we added a CH3 to it. We said that that was a branched chain, right? Well, branched chain, we had this 1 carbon sticking up, that's our CH3 here. Remember, 1 inorganic is meth, so we call this a methyl group. Instead of the A-N-E on the end, we put a YL to show that it's a branch on something. And this is a general nomenclature that you use a lot in organic chemistry. It's methane. Here's our methyl group, so it's attached to something else. For example, methyl alcohol. There's our CH3. The alcohol functionality is an OH. Methylamine. Here's our CH3, our methyl group. The amine is a nitrogen with 2 hydrogens. So this is just a general sort of nomenclature. Again, it describes the number of carbons that are attached as a branch to something else. Just for an example, again, 1 carbon, this would be methane, and we call it methyl. 2 carbons, ethane, ethyl. 3 carbons, prop, propyl, butyl, pentyl, whatever. So you just name it based on the number of carbons that are stuck onto something else. Now, the diversity of carbon compounds is what makes organic chemistry so incredibly fun. I teach organic all the time, taught it for 30, 40 years. It's a great subject if you ever manage to get there. This is a branch chain alkane, isn't it? Here's our zigzag, and we have a methyl group sticking up. Here we have 6 carbons in a ring. So we can form not just straight chains, but we can form this ring. If you note, each of the carbons in this ring is tetrahedral, isn't it? So this is a cycloalkane. This is a bicyclic thing. So here we have 1 ring, another ring going up, a 3rd ring going down. The way you can just attach these carbon molecule or carbon atoms together is astounding. We can also stick in things like oxygens and nitrogens. We can make multiple bonds. This is a carbon-carbon double bond. We'll see those in just a little bit. This is glucose, the sugar. We have a ring here. We have 5 carbon atoms in this ring and 1 oxygen. On each of the ring carbons here, we have an OH group, that's an alcohol. Here's another one. And we have a CH2OH sticking up here. This is morphine. Morphine has this ring of double bonds. Again, here's another ring down here and another bridging ring up there. OH group, nitrogen in the ring down here. Very, very complex molecules. And each of these has very unique properties and reactivities. That's just one of the amazing things about organic chemistry. Well, if we have such very complex molecules, one of the great challenges is to find a way to represent these. Clearly, we could use models, as we've done here. This is a classic ball and stick. Again, This is where our zigzag comes from. If we do away with the little balls and just keep the sticks, this actually has a name. It's called a drying model. This is a space-filling version of each of these. These are all on the same scale. In a space-filling model, we extend the size of the little globes so that they actually represent more or less where the electrons are. So this is what the molecule really looks like. Problem with this is that it's difficult to see the structures, and so that's why we do these as well. None of us is such a good artist that we could ever reproduce one of these freehand, however, and therefore we need to find other ways to represent our molecule. This guy has 4 carbons. It has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 hydrogens. So this is C4H10. This would be butane. Remember, 4 is a butte. If we simply wrote the molecular formula, we could look at this and say, yeah, that's an alkane, and could be butane, could be other things too. We'll see that in a minute. So the molecular formula tells us the atoms that are there, but it doesn't tell us very much about the molecule itself. This is what's known as a condensed structure. In a condensed structure, you can convey the information of how these things are bonded together. And this is actually fairly useful. We have a CH3 on the end. We have 2 CH2s, these guys, and a CH3 on this end. When you draw a condensed structure, you are expected to know that all the carbons are bonded together, just like they are here. Another way to draw this is called a Kekulé structure. In a Kekulé structure, you actually show all of the atoms, all of the bonding, with our little dashes to represent the covalent bonds. This is very useful, except that it gets to be a real pain. Organic chemists get real tired of drawing little H's and stuff like that. Therefore, this is the ultimate in simplicity, and this is what we use mostly when we draw organic compounds. This is called a line or a structural drawing. Anytime you have a line just going off into space and stopping, that's a truncated line. That's a CH3 group. So this guy here represents this atom here. So that's a CH3. Anytime you have a vertex here, that's our zigzag, that's our CH2. So we would look at this structural drawing, line drawing, and we would say CH3, CH2, CH2, and a CH3. Virtually all organic compounds that you see will be drawn using some type of a structural drawing. So once again, this is our basic conversion from the Kekulé here. Again, these are CH3s, these guys are CH2s, and another CH3. This is a pure or a strict line drawing. However, sometimes, just to make it easier on you, chemists will take and they'll show these CH3s here by just writing them out like that. That makes it a little easier to interpret. Again, these are CH2s. Sometimes people will look at this and say that looks odd. Looks like you're saying that the hydrogen is bonded down here, so they'll reverse them. So you can show that the carbon is attached. In reality, when you see organic structures drawn, there'll typically be a mixture of all of these, from the very pure to something like this, to the thing that's almost right. And you're just supposed to more or less be able to look at them and figure them out. So here's our basic structural or line drawing. And we've shown our carbon as our point of attachment. Another form that you'll see sometimes is called a sawhorse. In a sawhorse projection, same molecule, here we're going to show all of our atoms. And what we're doing is just taking this carbon and putting it in the front. So here's our CH3 sticking up. This carbon has 2 hydrogens, so we do that. And I suppose if you flip this other guy over, it would look like a sawhorse, and that's where the name comes from. The one that's most confusing, and of course that we see all the time, is what's called a Newman projection. Same molecule, all in the world you're doing with a Newman projection is you're looking straight down this axis here. So this is our front carbon, that's this guy, with the CH3, 2 hydrogens, CH3 is 2 hydrogens. Our back carbon we draw as the big ball. So you know it's in the back, again, methyl group and 2 hydrogens. The process of going from this to this, I made a little movie just to kind of show it. Here we start off with our drying model. Okay, it's supposed to run. There it goes. Now we're at the sawhorse. And finally, this is the Newman. This is our back atom, and it's nice and big. Once again, here's our guiding model, the sawhorse, and then finally our Newman. All right, let's just go ahead and do a very simple set of problems here. Let's take these line drawings, calculate drawings, condensed structures, or whatever, and let's convert them into pure line drawing. It's kind of a fun exercise. I will pause here for a moment while you ponder these things. For our first guy here, we have a condensed structure. When you have a branch on a condensed structure, you show the branch here in parentheses. So the first thing that you want to do when you're converting from this to a line drawing is to figure out how many carbons you have in your basic line, okay, in the basic structure. So this is going to be 1 here. This is 2, 3, 4, and 5. These 2 methyl groups, in parentheses, are branches. And the way the nomenclature works, it says that both of these methyls are attached to this carbon. All right, so step 1, we have 5 carbons in a row. We're going to draw our zigzag. And it's going to look like that. We're doing 1, 2, 3, 4, 5 carbons. Over here on carbon number 2, we have 2 CH3 groups. So we'll go and find carbon 2 and simply put in 2 lines, remembering that each of these represents a CH3. So we interpret this as a CH3, 2 more CH3s, and then 1, 2, 3 carbons in our zigzag. Our next guy, we have 6 carbons in a ring. 1, 2, 3, 4, 5, 6. These are all CH2s, aren't they? Remember what a CH2 is in a line drawing. It's just a vertex. So all in the world you have to do is convert this from this calculate form with all these hydrogens into something like this. Each of these represents a CH2, and this is our basic zigzag, up, down, up, down, up, down. This is referred to as a chair conformation. The molecule itself is called cyclohexane, and this is chair cyclohexane. The organic chemists who have imaginations. You can look at this and imagine it is a chase lounge or something like that. So you're sitting in this nice little comfy chair. This guy is a little more difficult to take apart. It's a Newman projection, isn't it? What you want to do is to figure out how many carbons you have in your chain. So we're going to do 1, 2, 3, this is our front carbon. 4 is the back carbon, 5, and then 6. So we need a zigzag with 6 carbons. On carbon number 1, 2, 3, we have a methyl side chain. So 1, 2, 3, methyl side chain. On carbon 4, we also have a methyl side chain, and that's this guy. Now I've shown this stepwise. Sometimes if you have a Newman and you want to untangle it, it's easiest just to take it back to our sawhorse. And now you can see, here's our main chain, 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. And we have 2 methyl groups on carbons 3 and 4. 3 and 4. Any questions? All right, well, carbon compounds like these, simple alkanes, all have single bonds. And because they have single bonds, you can rotate them, rotate everything around each other. These two are the same molecule. And all we've done to convert one to the other is to simply rotate around these single bonds. We rotate, we could make this. Carbon compounds rotate rapidly. In fact, so rapidly, like 10 to the 6 times per second, that they are just extremely dynamic. We call these conformational isomers. They look different, but they're the same molecule. Here's a little movie that I made that kind of demonstrates this. Again, because the rotation is so fast, we regard these as the same molecule. So basically, if you were to draw this molecule, you could draw it this way or frankly any other way, and you'd still be right. Here's our movie. This is a pentane, and it's just here doing gymnastics, isn't it? This is just supposed to represent exactly how many degrees of freedom we have around all of these bonds. Another way to look at it, these are all conformational isomers. We have 1, 2, 3, 4, 5 carbons in a row, and we have a methyl group here on our carbon number 3. There's our main chain, and here's our side. Well, this looks a little different than that one, but again, here's our 5 carbon main chain and our methyl group on carbon 3. Here we've just tipped it down like that, still a methyl on carbon 3. So carbon compounds are incredibly dynamic. they can adopt any conformation. And I'm not really going to ask you to draw carbon compounds, but basically, if you were to draw something like this as a line drawing or whatever, any pathway that you wanted to put, again, would be acceptable because they just interconvert so incredibly rapidly. All right, rather than just talk about compounds in general terms, let's go ahead and just look a little bit at how we actually name these things. Once again, on your exam, you will not have to name compounds specifically. But let's just go ahead and go through the exercise. Kind of a fun exercise. The first thing you do is you want to look at your molecule and you want to identify the longest continuous chain of carbons. Now for our first one here, that's very simple. It's going to be our zigzag here, 1, 2, 3, 4, 5, 6 carbons. And this would look like that. Over here, our longest chain. Well, probably we want to start up here, 1, 2, 3, 4, 5, 6. On this side, 1, 2, 3, 4, 5, 6, 7. Either go up or down, doesn't matter. So that's our first thing. Once you identify the longest chain, we call that the parent. Now remember how we talk in organic chemistry. We use the prefixes to indicate the number of carbons. So this has 1, 2, 3, 4, 5, 6. Remember 6 was hex. It's an alkane, so this is hexane. Over here our parent also has 1, 2, 3, 4, 5, 6. So this is a hexane parent with a methyl group. 1, 2, 3, 4, 5, 6, 7. This is a heptane with an ethyl and a methyl. So that's how you're going to name it. Let's look at this molecule. First, we want to find our longest chain. The longest chain, well, we're going to go this way or this way. Let's see. If we go along the bottom here, we have 1, 2, 3, 4, 5, 6, 7. So our parent would be a heptane. We can also look at it this way. 1, 2, 3, 4, 5, 6, 7. That's the same heptane, isn't it? So there are two ways you could represent the longest chain. Again, these are just simply conformational isomers. Once we look at the thing, we want to try to pick out the chain, the parent chain here, that has the greatest number of substituents. So here we have 1, 2 side chains, here 1, 2 side chains, they're the same. We can also make this 7-carbon unit our parent, but we would only have one side chain. So this one is preferred over this one. Now, once you have identified your parent chain, in order to name an organic compound, you need to assign numbers along the chain. What we're looking for here is to start at one end of the chain so that we get the lowest numbers we can. If we started here, this methyl group would be in carbon 2. If we started here, our first substituent would be in 1, 2, 3, 4. So starting here is a better way. That will give us lowest numbers. All right, so let's take this and let's name it. Step 1, we want to look and try to find our longest chain with the greatest number of substituents. The longest chain here, I think, would look something like this. 1, 2, 3, 4, 5, 6, 7, 8. So this would be an octane. 8 is oct. I started numbering down here with this carbon because our methyl groups here are going to be in carbon 2 that way. If we started numbering here, our first substituent would be in carbon 3. So 2 is better than 3. Just keeping track of what we have here. We have 2 methyl groups in carbon 2, another one over here in carbon 6, and we have a 3-carbon side chain that's a propyl in carbon 1, 2, 3, 4. So keeping score, we have 2, 2, 6 trimethyl, 2, 2, 6, and 4, we have a propyl. Finally, when you put all these things together, you need to indicate your main chain. That will be our last word. And we just put these guys alphabetically in front. The tri doesn't count, the methyl does. So the actual name would be 2,2,6 trimethyl 4-propyl octane. 2,2,6 trimethyl 4-propyl octane, all one word. Now we're not going to have to do that on an exam, but again, if you see an organic name like this, going down to this end, that tells you your parent chain, 8 carbons, and this just describes where along that parent chain you have substituents, and of course, what they are. Any questions? Let's look at one more here. Here, instead of a hydrogen, I'm going to stick a chlorine on. Now this makes a functional group called an alkyl halide. We'll talk about those in a bit. But here's our basic 8-carbon chain. Instead of a methyl here, we now have 2 methyls in carbon 2. We have our propyl still in carbon 4. And over here we have a chlorine in carbon 6. So octane is our parent, 6-chloro-2,2-dimethyl 4-propyl. Any questions? Let's just look at these and give a couple very simple names here. Once again, we're dealing with a Newman. We all hate that. Let's untangle this Newman and try to find our longest chain. We're going to have 1, 2, our back carbon is 3, 4, and 5. So it looks to me like a pentane. Drawing this as a sawhorse. Again, there's our front carbon. 1, 2, 3, 4, 5. So we're dealing with a pentane. On this pentane, so here's our parent chain, we have a methyl group, and that will be a carbon 1, 2, 3. This is quite simply, here's our parent. Start numbering, one end or the other, doesn't matter. And we have 3-methyl pentane. This one's a little simpler. Our parent is going to look like this, 1, 2, 3, 4. So it's a butane. We want to start numbering down here. And we have 2 methyl groups in carbon number 2. So here's our 4-carbon chain. Again, doesn't matter which one you choose. And 2 carbons in carbon number 2. So this is carbon 1, that's 2, 2,2-dimethyl butane. Now if we had changed this and stuck a couple chlorines in, Same sort of thing. We want to find our longest chain. Here we're going to let this be part of our longest chain, so the chlorine here can be on carbon 1. That would be the lowest number. We have a chlorine here in carbon 1, another one in carbon 3, and 2 methyls in carbon 2. So that's 1. Put them all together. 1,3-dichloro, 2,2-dimethyl, and again our parent is a butane. Now in organic chemistry, you also do things backwards. And this is even simpler. We look at this guy. We have a butane as our parent. So that's 4 carbons. Let one end be carbon 1. Now we're going to have 3 methyl groups at carbons 2, 2, and 3. So this is carbon 2, that's carbon 3, and there's our 3 methyl groups. Isn't that simple? Hexane, so we have 6 carbons in a row. Let one end be carbon 1. On carbon 3, we have an ethyl, so that's a 2-carbon side. At carbon 2, we have 2 methyls, that's here, and that's our molecule. Finally, down here we have octane, so that's 8 carbons in a row. And my goodness, we have methyl groups here at carbons 2, 3, 4, 5, 6, and 7. That's darn near all of them, isn't it? And it would look like that. So organic chemistry, very systematic. You can have very complex molecules, name them very simply, and everybody in organic chemistry, of course, understands what you're talking about. There is one clarification I want to make here, though. When we talked about conformational isomers, we said these things are spinning around really, really fast. They spin at roughly 10 to the 6 times per second. And that's why we don't really regard these as unique compounds. It would be simplistic, however, to say that they had identical energies. If you want to think about it, here we have these 2 chains fairly close to each other, don't we? Here they're as far apart as they could get. This has got to be slightly higher energy than this guy. So just statistically, you're much more likely to be in the lower energy than in the higher energy. This can be shown with a very simple compound. This is ethane. And here's a little movie. And as we rotate around our single bond here, you can see these hydrogens get really close to each other, don't they? That's a higher energy state when they're up here in between. So like this one, called a staggered conformation, that's the lowest energy. We can actually look at a plot of these energies, and as we rotate around our bond here, we see that this changes about 12 kilojoules per mole. So it's a real difference in energy, but once again, conformational isomers we're going to regard as being the same molecule. Now, there are other types of isomers in organic chemistry, but these are fun. And these are called constitutional isomers. If we look at these 2 guys, here we have 1, 2, 3, 4, 5 carbons in our parent chain and a 2-carbon side. Here we have 1, 2, 3, 4, 5, 6 carbons in our parent and a 1-carbon side. But This is C7H16, and so is this. They are clearly different molecules. We call these isomers, and these are constitutional isomers because the constitution, the backbone, is unique among both of them. With carbon compounds, again, you can have huge numbers of constitutional isomers. Now, one of the really fun games that you can do in organic is to do this, and that is write all the constitutional isomers having a given formula. So if I told you we had C3H8, the only way you can arrange C3H8 is to do this simple propane, the only possible isomer. Here it is this way. Go ahead and see if you can draw a C3H6. How many C3H6s can you make? The way you want to do it is just to start off with 3 carbons, connect them. You've only got 6 hydrogens now to connect them together. Now you need to remember that carbon can form single bonds, it can form rings, and it can form double and triple bonds. So our isomers can be any combination of those. The only ways that you can do C3H6 turn out to be these 2 compounds. We can actually take these 3 carbons, wrap them in a triangle. This is called cyclopropane. Very high energy molecule because we're bending these bonds way back. And here we are making a carbon-carbon double bond. Again, this is 3 carbons, 1, 2, 3, 4, 5, 6 hydrogens. Once again, look at this, the valence of all these carbons is still 4. Here we have 4 bonds, 1, 2, 3, 4. Here we have a double bond, so that's 1, 2, 3, 4. 1, 2, 3, 4. 4 bonds to all the carbons. Double, single, triple, rings, etc. C4H8. Well, here we're going to have more possibilities. The assortment looks something like this. Once again, 1, 2, 3, 4 carbons. 1, 2, 3, 4, 5, 6, 7, 8 hydrogens. Same thing here. All of these are constitutional isomers. Made our ring here, 4 carbons in a square. Here we have our 3-membered ring, and I just put 1 carbon sticking out as a side chain. Let's interpret this. We have 1, 2, 3, 4 carbons, 3 hydrogens, 4, 5, 6, 7, and 1 hydrogen here. That's 8. Part of the art of organic is being able to look at these different structures and come up with an understanding of what the molecule really looks like. Here I just made it just a little more complicated just to show you the diversity. If we stick in a couple oxygens, you would think that that's a fairly minor change. However, these are all valid organic molecules that are C3H6O2, and so are these. So organic chemistry, the molecules can get very, very complicated very, very quickly. Now you will get a simple question on your exam about conformational or constitutional isomers. So let's make sure that we understand this, okay? Question is going to look something like this. You'll be given a couple compounds and you will be simply asked, are these identical, constitutional isomers, or different compounds that are not isomeric? Now, how do you do that? The simplest way here is just to look at your structures and come up with a formula. So, here we have 1, 2, 3, 4, 5 carbons, right? So this is going to be C5. Hydrogens, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. This is C5H12. Now, let's look at this guy. We have 1, 2, 3, 4, 5, 6 carbons in a row. 3, 6 hydrogens, 6, 7, 8, 9, 10, 11, 12, 13, 14, C6H14. In order for these guys to be isomers, they have to have the same formula. Therefore, these guys are different compounds and they are not isomeric. Go ahead and quickly do this one on your own. Step 1, you want to do the formulas. Once you have the formulas, decide if they are in fact the same molecule, isomers, or if they're different. Interpreting our structures here. We have 1, 2, 3, 4, 5, 6 carbons. So there's C6. Add up the hydrogens. Well, let's see, we have 2 chlorines also. And adding up our hydrogens. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 6 carbon main chain. We have chlorines in 2 and 4. Oops. I thought I had the formula on here. Let's go over here. Our main chain is going to be 1, 2, 3, 4, 5, 6 carbons. We have a chlorine here in carbon number 2. Here we have a chlorine in carbon 2. We also have a chlorine in carbon 4, so this would be 1, 2, 3, 4. Same thing here. They both have the same number of carbons, the same number of hydrogens, and chlorines. These guys are the same molecule. So we will have a question, like I said, where you're going to have to look at a pair, interpret the structures, and decide identical, isomers, or different. Any questions? Yeah?
So right away when looking at the carbons, you would know if they were similar or not, right? Because like if one had 4 and one had 5.
If you simply did the formula here, you would know, this is C6, I forgot how many hydrogens, chlorines there were, but if we did this formula, and this one would be the same. Now that tells you right away that they're either identical or isomers. Then you just kind of look and say, oh, okay, we have chlorines, 2 and 4, chlorines, 2 and 4. They're the same. All right, let's do a little bit more about our double and triple bonds and where they actually come from. These are what we'll call functional groups in just a minute. Anything with a carbon-carbon double bond, we call an alkene. E to indicate the double bond. Anything with a triple bond, we call an alkyne. So this has 2 carbons, so it's an F. It's an alkyne, so we call this ethyne or ethene. Now, these double and triple bonds are actually very important things in organic chemistry because they make these molecules very reactive. In organic chemistry, you want to do lots and lots of reactions. To build a double bond, we call this an sp squared hybrid. So we're going to take the electrons from our s and 2 of our p electrons, and we're going to put them together and build a trigonal structure like this. These are the real bonds. Because we have 3 things, this is trigonal and it's planar, 120-degree bond angles. But remember, we had 1 p orbital left over on each carbon. We've used 3 of them to make our backbone, but we still have 2 p orbitals left over, 1 on each. And they actually look like that. To make our double bond, what we do is simply allow these orbitals to overlap. And they form what looks like a hot dog. So here's our bun. And the sigma structure down here is buried in the middle. This is our double bond. One of them is our real bond, and the other bond is this, we draw it just as a real bond, but it's actually this cloud of electrons above and below this backbone. This is a very reactive sort of arrangement. If we look at the electrostatic potential, you'll see all of these electrons show up as these bright red spots. And these are the things that give organic molecules reactivity, because we have rich regions of electron density. This also makes this structure rather rigid, because this is a single bond, right? And we saw that we can rotate around single bonds. But in order for these guys to overlap, to form our hot dog bond, if you will, they must be exactly aligned with each other. If we rotated it so it was like this, these would no longer overlap, and so we cannot rotate around this bond. Perhaps you have heard of cis and trans fatty acids. This is an example of a cis, where you have things on the same side of a double bond, and this is a trans, where they're on opposite sides. Cis and trans simply refers to the fact, because you can't rotate around this bond here, these things are frozen in space in either the same side or the opposite side. In terms of fatty acids, cis fatty acids occur naturally. They're easy to metabolize. Trans fatty acids do not occur naturally, and your body cannot metabolize them. So it just stores it under hips, okay? In organic chemistry, you would call these things stereoisomers. For the sp center, Again, we're going to triple bond. We're going to use 2 orbitals to make our sigma network. We have p orbitals left over, 2 on each. And here we have linear geometry with 2 sets of these pi orbitals. Again, very reactive geometry. Each of these we draw as a bond like this, that's our triple bond. And as you can see, we have a huge belt of electron density around the middle. So in terms of functional groups, our alkane, all carbons are sp3 hybridized. Alkenes, we have double bonds. Alkynes, we have triple bonds. Now again, these are examples of what we call functional groups. The thing about functional groups that makes organic easy is that you don't need to worry about the details of the molecule so much. All you do is you simply look and say, what functional group is in there? Here we have a double bond. So this is an alkene. Here we have an OH. This will be an alcohol. This is an oxygen in the middle, called an ether. All of these have very unique reactivities that are actually independent of what all is attached to them. If we take any alkene, so any carbon-carbon double bond, and we react it with bromine, we always form a 1,2 dibromide with the bromines opposite or trans to each other. So again, this is the trick to learning organic. You don't really need to worry about what's attached here. You just look at the fact this is a double bond, and this is the product you always get with that double bond. Now, this is actually the mechanism of it. This is our cyclic double bond here. This is bromine. What we do is form this bridging intermediate called a bromonium ion. This then reacts with the other bromine now, ring opens, and then we get our trans 1,2 dibromide. Organic chemistry, you worry a lot about the mechanisms, the pathways of the reactions. Just to give you an idea, this is a nice carbon-carbon double bond. In organic, we would talk about all of these reactions and more. So there's a lot in there, but again, they're all simple functional group reactions. Let's just very quickly run through our functional groups. Again, you're not going to be held responsible for this, but it's important. Alkanes, we said, only sp cubed. Alkene, double bond. Alkyne, triple bond. If we put 6 carbons in a ring with 3 double bonds, this is an arene. That's a benzene. Now, you may have heard of benzene. Benzene is a very, very good solvent. When I was an undergraduate, we used to wash our hands with it. Now it is considered a deadly carcinogen, so can't do that anymore. It also showed up in traces in like Perrier drinking water. When they pulled it out of the ground naturally, it had benzene in it. So they finally had to scrub it all out. Our benzene actually looks like this. These are all the p orbital left over. And this is the electrostatic potential map. You can see the big cloud of electrons above and below our plane. Benzene reactions are very important in organic chemistry. Put a halogen on, call it an alkyl halide. OH is an alcohol. This is an ether. We saw one of those. Put a nitrogen out here. That's an amine. This is carbon-nitrogen triple bond like cyanide. That's a nitrile. Nitro group, SH, that's a thiol. The essence of skunk is a thiol. Very, very, very smelly compounds. Sulfur in the middle is a sulfide. And finally, we have whole family that feature carbon-oxygen double bonds, aldehydes, ketones, carboxylic acids, esters. Amides are important, we'll see those in a minute, acid halide, anhydrides, etc. The reason the carbonyl is so important is that this is a very, very polar bond. This is acetone, carbon-oxygen double bond. It sucks the electrons up towards the oxygen, like this, to make a very, very polar compound. That means this is a positive carbon, and it can undergo reactions like this. Things with electrons can add. When they do, you can form addition intermediates and go on to make different types of compounds. This is the sort of stuff that you do in organic chemistry. Now, I said that amides were important. Remember, an amide was a carbonyl with a nitrogen on this side. These are important because these are the things that show up as peptide linkages in proteins, and we start with amino acids. This is an assortment of amino acids. These are all positive. Again, you can note our carbon structure here. This is a carboxylic acid, an amine. These are all nitrogens. Here's a cyclic amine. All of these are different amino acids. Here are uncharged side chains. This is thiol down here. This is glycine, that's the simplest. Proline, that occurs in hair, stuff like that. And the final set, these are hydrophobics. Again, we have benzene rings in these guys down here. There are 20 naturally occurring amino acids. These naturally occurring amino acids bond together to make proteins. The way they do that, you'll notice that all of these have a carboxylic acid and they have an amino group, right? If we take one amino acid and another, we can react this carboxylic acid with this nitrogen, and we're going to make an amide. When we do this, we've made what's called, in biochemistry anyway, a peptide bond. It's nothing more than an amide right here. Now, this end of the molecule, we have another carboxylic acid. This end, we have another nitrogen. So you can actually continue this chain by sticking other amino acids in. This is a leucine, a glycine, a serine, and an alanine. Put these all together, and that's called a peptide. We make a peptide really long, and we call it a protein. These are, again, all of the amino acids, and they're simply bonded by these amide or peptide bonds. So, once you make an amino acid polymer like this, because all of the side chains are so unique, they have different water solubility, etc., this chain will spontaneously fold up. And it'll fold up into a very, very unique structure. This is the enzyme lysozyme. Again, this is nothing more than our polyamino acid backbone, but as you see, different regions will coil up. Here we have what's called a beta sheet in between. These are loops and stuff like that. The structure of this protein is fixed by the number and types of amino acids. When an enzyme performs a chemical reaction, this very unique structure that it forms is such that it can bind specifically in some of these crevices a very specific molecule, and it does the organic chemistry on it. Very, very amazing. This is the same molecule here. I've just shown it now as a space-filling model. Again, very unique structure, and they all basically look the same. All lysozymes will look exactly like this one. Let's go to DNA and RNA. We all know the initials. What does the D stand for? Well, they all have a backbone of sugars. The 5-carbon sugar here, 1, 2, 3, 4, 5 up here, this is called ribose. Oxygen bridging these 2 carbons here. Again, OH, OH, CH2OH. The difference between ribose and deoxyribose is simply that we have an OH group here on carbon 2. Here we have a hydrogen instead. So that's the D that's in DNA. In RNA, you have ribose only. DNA, you have deoxyribose. That's enough of a difference to set the two totally apart. These are 2 of what are called DNA bases. This is guanine and adenine. Again, if you look at the structures, these are all line drawings. We have nitrogens in our rings up here, carbon-oxygen double bond. This is our deoxyribose. Same thing here. For here, we have a slightly different base up here. Instead of a carbonyl, we have an NH. And we also have an NH2 down here instead of a hydrogen. These are the other 2 bases called pyrimidines. Again, deoxyribose down here. Up here we have cytosine, so an NH2, carbon-oxygen double bond, 2 carbonyls, and an NH. This methyl group is not present in RNA, where we have uracil as our base instead of thymine. Now, as we said when we talked about hydrogen bonding, the thing that makes DNA work is that we can take these bases and line them up. And we can form hydrogen bonds between these functional groups on the sides. So for a GC pair, we have 1, 2, 3 hydrogen bonds we can form. This means that a G will always and only pair with a C. And that's how the DNA alphabet works. If you look at our pair here, here's a GC pair. You see the poly sugar phosphate backbone on both sides. And here's the AT pair, and here's the GC pair. These all bind together appropriately and then wind up to make our double helix. The double helix looks something like this. Again, here's our backbone, and here's all of our pairs bonding with each other. Kind of a beautiful sort of arrangement. All right, so we've done proteins. We've looked at nucleic acids. The last thing I want to talk about, oh, here it is in a very nice space-filling model. Again, you can see the ribose phosphate backbone. And down in the middle here is where we have all these base pairs hydrogen bonding to each other. So let's do fats. Let's wind up with fats. This is a double bond, isn't it? This is a cis double bond. Remember, cis is natural. This is called a fatty acid. Carboxylic acid on this end, long chain of carbon atoms here with a double bond. We can make what are called triglycerides if we take 3 fats and stick them together. This is actually esters, ester bonds, so carbonyls. And we do this with glycerol, so it's a trialcohol. These things are the things that are used to build membranes. We have a very, very hydrophobic fatty end here. And on this end, we can put groups that are hydrophobic, or hydrophilic, I'm sorry. So here we have an example, 2 long chains. This is charged out here. This is called choline, positive nitrogen. When these things self-assemble, all of these fatty chains tend to bond to each other. The charged groups here all migrate up to the surface. This is a membrane. And this is the thing that surrounds all the cells, etc. Basically keeps an inside and an outside, totally enveloping everything inside the cell. We can't very well talk about fats without talking about everybody's favorite thing. This is cholesterol. Now that you know organic chemistry, you can interpret this structure. Again, we have rings. These are all CH2s, CH3s, some hydrogens here, and an OH group. Cholesterol, because this is very water insoluble, will embed itself in that membrane that we just saw, with the OH, the hydrophilic part, sticking out towards the top. Cholesterol is something that everyone is familiar with because, and here it is as a ball and stick model. Again, this is the water-soluble end, this is the insoluble end. But these things can accumulate within membranes, etc., and form what are called arterial plaques. So here is lots of cholesterol-laden fat built up inside of somebody's little artery. The actual mechanism that this follows as plaques form is fairly complicated. Cholesterol kind of has a bad name. People will tell you that you need to eat low cholesterol this and low cholesterol that. We'll talk about food in another chapter, but that's actually not right because your body makes cholesterol like mad. Your liver is just a machine for making cholesterol. Most of the cholesterol that's in your bloodstream is actually made by your liver. The stuff that you get when you eat eggs doesn't touch it. So the mechanism by which cholesterol gets embedded in these plaques, how they form, is not quite that simple. It's not this that you eat fat, you eat cholesterol, and you get plaques. No, there's much, much, much more to it than that. All right, well, this chapter was just supposed to be an introduction, an overview of organic chemistry, so you can look at an organic structure and have a feel for what it really is carbons and hydrogens and stuff like that. And finally, linking these together into the biomolecules that we use to make proteins, DNA, and finally fats. Any questions? All right. Remember, no lab on Monday. That's number one. Number two, there is an attendance sheet somewhere. If you have not, there it is. If you have not signed it, please do. And number three, we are at the point where we're going to have our second exam. So the next lecture we have will be our sample exam. And after that, then we will have the real